Hello, dear students. In this video, we are going to learn about Mendelian genetics. At the end of this lesson, you will be able to describe Mendel's experiments and results, use a Punnett square to perform monohybrid crosses, and explain how alleles influence inheritance. This is this lesson's essential question. How can we use the laws of inheritance to predict the genotypes and phenotypes of parents and their offspring? Genetics is the scientific study of heredity and variation, where heredity refers to the transmission of traits from one generation to the next, and variation explains why offspring are similar to, but not identical to, their parents or siblings. Before formal laws of inheritance were established, scientists proposed two main ideas. First is preformation, which claimed organisms existed fully formed in miniature within reproductive cells. Second is blending inheritance, which suggested parental traits mixed together during conception. Gregor Mendel showed through his pea plant experiments that traits are inherited as distinct units, now called genes, rather than blending, resulting in predictable patterns of inheritance across generations. The diagram illustrates Mendel's experimental design for controlled cross-pollination in pea plants, which allowed him to study inheritance accurately. Pea flowers normally self-fertilize because the male anthers and female carpel are enclosed within the same flower. To prevent this, Mendel first removed the anthers from a purple flower, eliminating its ability to produce pollen. He then collected pollen from a white-flowered plant and carefully transferred it to the stigma of the purple flower. This ensured that fertilization occurred only between two plants with known traits. When the seeds from this cross were planted, all offspring produced purple flowers, showing that flower color did not blend but followed a predictable pattern. By controlling which plants acted as pollen donors and recipients, Mendel was able to trace how individual traits were passed from one generation to the next. The figure summarizes the results Mendel obtained from crossing pea plants with different flower colors. When he crossed pure-breeding purple plants with pure-breeding white plants, all offspring in the first generation produced purple flowers, showing that the purple trait did not blend with white. When these first-generation plants were allowed to reproduce, the white flower trait reappeared in the next generation, where about 3/4 of the plants had purple flowers, and 1/4 had white flowers. From these consistent results across many crosses, Mendel concluded that traits are passed on as separate, stable units rather than blending together. Mendel examined 7 pea plant traits, such as stem height, flower color, and seed shape, distinguishing dominant traits, which appear when at least one dominant allele is present, from recessive traits, which appear only when 2 recessive alleles are inherited. In the F2 generation, he consistently observed an approximate 3-to-1 dominant to recessive ratio. Based on his experimental data, Mendel described consistent patterns of inheritance in pea plants. Scientists later formulated the following laws. The law of segregation, which explains that allele pairs separate during gamete formation. The law of independent assortment, which states that different traits are inherited independently of one another. And the law of dominance, which states that a dominant allele masks a recessive one in heterozygous offspring. Mendel presented his results in 1865 and published them in 1866 in experiments on plant hybridization. His work received little attention at the time, and Mendel himself was cautious about its broader applicability, suggesting that similar experiments would be needed in other species. This occurred shortly after the publication of Darwin's On the Origin of Species, which Mendel had read, although Darwin was unaware of Mendel's work. The diagram explains natural selection by showing that populations overproduce offspring, individuals vary in traits, and those with advantageous variations are more likely to survive and reproduce, passing those traits to the next generation. Over time, these inherited traits become more common, leading to evolutionary change. Darwin observed variation and inheritance and proposed the theory of natural selection without knowledge of genes. Mendel's later work revealed the mechanisms of inheritance, demonstrating how traits are passed from parents to offspring. Together, Darwin's theory of natural selection and Mendel's discoveries laid the foundation of modern evolutionary theory. Gregor Mendel's contributions are impressive because he described basic patterns of inheritance before genes and chromosomes were discovered. This timeline shows key discoveries in genetics, starting with Mendel's work on heredity patterns in pea plants in 1865, leading up to genome sequencing projects in the 1990s. After Mendel's discoveries, scientists clarified how traits are expressed through allele combinations. Each gene occurs in pairs on homologous chromosomes, with different versions called alleles. A cell is homozygous when it has 2 identical alleles, and heterozygous when it has 2 different alleles. In traits showing complete dominance, a dominant allele is expressed whenever it is present, while a recessive allele appears only when both alleles are recessive. Genotype refers to the genetic makeup of an organism for a specific trait. It describes the particular combination of gene versions inherited from the parents. In this example, flower color is controlled by a gene that has two versions, one that produces purple flowers and one that produces white flowers. An allele is a different version of a gene. An organism can inherit two identical alleles, which is called homozygous, or two different alleles, which is called heterozygous. When both alleles are the same, they may be represented as two capital letters, or two small letters, meaning they give the same genetic instructions. When the alleles are different, they are represented by one capital letter and one small letter, and the allele shown by the capital letter usually masks the effect of the one shown by the small letter. The phenotype is the observable trait produced. For flower color, purple appears when at least one purple allele is present, while white appears only when two white alleles are inherited. As a result, most offspring show purple flowers, demonstrating how genotype determines phenotype and how dominance affects trait expression. Now let us focus on what Mendel concluded from his experiments and what his laws mean. The law of segregation states that an organism has two alleles for each trait, and these alleles separate during gamete formation. This separation occurs during meiosis, so each gamete receives only one allele for a trait. When fertilization happens, the offspring receives one allele from each parent, restoring the pair. Mendel discovered this by observing that traits that disappeared in one generation could reappear in the next, showing that alleles do not blend but remain separate units of information. The law of independent assortment states that the inheritance of one trait does not affect the inheritance of another trait, as long as the genes are not located close together on the same chromosome. The law of dominance states that in a heterozygous individual exhibiting complete dominance, the dominant allele is expressed while the recessive allele is masked. This pattern of inheritance is commonly illustrated using a Punnett square, which shows how dominant and recessive alleles combine in offspring. The Punnett square is a diagram used to predict the possible genetic outcomes of a cross between two organisms. It is widely used to visualize Mendelian inheritance. Let us consider this problem. In pea plants, yellow seed is dominant over green seed. Complete a Punnett square for a purebred yellow-seeded pea plant crossed with a purebred green-seeded pea plant. Step 1. Identify the trait and its alleles. Yellow seed color is dominant, while green seed color is recessive. Capital letter Y represents the allele for yellow seeds, and small letter y represents the allele for green seeds. Step 2. Identify the parents and their genotypes. The cross involves a purebred yellow-seeded plant, which has two identical dominant alleles, and a purebred green-seeded plant, which has two identical recessive alleles. Because they are purebred, each parent has only one type of allele to pass on. Step 3. Determine the possible gametes. Each parent forms gametes during meiosis that carry only one allele. The yellow-seeded parent produces gametes carrying the dominant Y allele. The green-seeded parent produces gametes carrying the recessive Y allele. These possible gametes are placed along the top and side of the Punnett square. Step 4. Fill in the Punnett square. Each box in the Punnett square is filled by combining one allele from each parent. Every box contains one dominant Y allele from the yellow parent and one recessive Y allele from the green parent. As a result, all offspring have the same allele combination. Step 5. State the results. Genotypic ratio, 4:0, or 100% heterozygous. Phenotypic ratio, 4:0, or 100% yellow seeds. No green seeds appear because the recessive allele cannot be expressed when paired with a dominant allele. Practice problem 1. Cross a heterozygous yellow-seeded pea plant with another heterozygous yellow-seeded pea plant. Consider the trait is Mendelian, and yellow-seeded pea plant is dominant over green-seeded pea plant. Step 1. Identify the trait and its alleles. The trait is seed color. Yellow is dominant, and green is recessive. Step 2. Identify the parents and their genotypes. Both parent plants are heterozygous, meaning each has one allele for yellow and one for green. Step 3. Determine the possible gametes. Each parent can produce two types of gametes, one carrying the yellow allele and one carrying the green allele. Step 4. Fill in the Punnett square. When gametes combine, 4 possible offspring combinations are formed. Any offspring with at least one yellow allele has yellow seeds. Only offspring with 2 green alleles have green seeds. Step 5. State the results. Genotypic ratio, 1:2:1. Phenotypic ratio, 3:1. Practice problem 2. A heterozygous round-seeded plant is crossed with a wrinkled-seeded plant. Give the genotypic and phenotypic ratio of the offspring. Consider that the trait is Mendelian, and the round seed is the dominant trait. Step 1. Identify the trait and its alleles. The trait is seed shape. Round is dominant, while wrinkled is recessive. Step 2. Identify the parents and their genotypes. One parent is heterozygous for round seeds, and the other parent has wrinkled seeds only. Step 3. Determine the possible gametes. The heterozygous parent can produce gametes carrying either the round allele or the wrinkled allele. The wrinkled parent produces gametes carrying only the wrinkled allele. Step 4. Fill in the Punnett square. When the gametes combine, two possible offspring combinations are formed. Offspring with at least one round allele have round seeds. Offspring with two wrinkled alleles have wrinkled seeds. Step 5. State the results. Genotypic ratio, 2:2. Simplified to 1:1. Phenotypic ratio, 2:2. Simplified to 1:1. Practice problem 3. Nearsightedness is a trait that is completely dominant over normal vision. Can a couple, both with normal vision, produce a nearsighted child? Step 1. Identify the trait and its alleles. The trait being studied is vision. Capital letter N represents nearsightedness as it is dominant. Small letter n represents normal vision as it is recessive. Step 2. Identify the parents and their genotypes. Both parents have normal eyesight. Since normal eyesight is recessive, each parent must have two recessive alleles. Step 3. Determine the possible gametes. Each parent can produce only one type of gamete because both alleles are recessive. Step 4. Fill in the Punnett square. When the gametes combine, all possible offspring receive one recessive allele from each parent. Step 5. State the results. Genotypic ratio, 4:0 or 100% homozygous recessive. Phenotypic ratio, 4:0 or 100% normal vision. The table illustrates common inherited traits in humans often used to explain dominance. Dominant traits include brown eyes, tongue rolling, free earlobes, Rh-positive blood, and dimples. Recessive traits include blue eyes, non-rolling tongue, attached earlobes, Rh-negative blood, and colorblindness. These examples help reinforce that dominant traits are expressed with just one dominant allele, while recessive traits require two recessive alleles. Mendel's law of dominance states that when two different alleles for a trait are present in a heterozygous individual, the dominant allele is expressed while the recessive allele is masked. Achondroplasia is caused by a dominant allele. An individual with heterozygous genotype will show the dwarf phenotype even though only one dominant allele is present. Albinism appears only when an individual inherits two recessive alleles. Parents with normal pigmentation can still have a child with albinism if both are carriers. To answer this lesson's essential question, we can use the laws of inheritance, such as Mendel's laws of segregation, independent assortment, and dominance, to predict the genotypes and phenotypes of parents and their offspring by analyzing the distribution of alleles during reproduction. To conclude, name one contribution of Mendel in genetics, compare genotype and phenotype, and explain how to use a Punnett square. Thank you for listening.